So plant pathology, ASRB NET 2006 question paper. So this question is also memory based and I had collected from my friends. So my name is NH Shankar Reddy and I'm doing PhD plant pathology in Annamalai University. So the first question is fungi and diseases in plants was written by E.J. Butler in 1918, whereas fungi and plant diseases was written by B.B. Munkur in the year of 1948. So these two books are very important and most of the times definitely we can expect at least one question from this. So fungi and diseases in plants was written by E.J. Butler. Fungi and plant diseases was written by B.B. Munkur. So Indian Phytopathological Society was established in there. Actually, it was established in the year 1947, February 28. But actually, it's a working condition. I mean, complete setup and working will starts in the year of 1948. So the next question is Indian Phytopathological Society was established by B.B. Munkur, who established the Indian Phytopathological Society, and antifungal antibiotic Ariofungin and Hyamycin was discovered by. So M.J. Tirumalacher is considered as father of Indian antibiotics. He discovered antifungal antibiotic Ariofungin and Hyamycin. So not only Ariofungin and Hyamycin, dermastatin, HYC4 and other more than 100 antibiotics are discovered by M.J. Tirumalacher. He is considered as father of Indian antibiotics, whereas Waxman is considered as father of antibiotics in entire world. But this Tirumalacher, M.J. Tirumalacher represents India. So antifungal antibiotic. Sometimes they will ask which of the following one is antifungal antibiotic. They will give options like Ariofungin, and then some of the antibiotics they will give. But make sure that Ariofungin is a very important antifungal antibiotic that was discovered by M.J. Tirumalacher. So the next one is the book Principles of Fungicidal and Action was written by J.G. Horsfall. It is also one of the important book. The Principles of Fungicidal Action was written by J.G. Horsfall. So the next one is Bodo mixture was developed by Milladet in the year 1882. So they will ask questions like this: Who discovered? Sometimes they can also ask like this: Who discovered Bodo mixture? Milladet. Composition of Bodo mixture 1:1:100, that is 1 kilogram of copper sulfate, 1 kilogram of lime or hydrated lime, or 100 liters of water. That is the composition of Bodo mixture. Actually, originally Bodo mixture was developed by Milladet in the year 1882. So the next question is Central Potato Research Institute is located at Shimla. So the next one is Morphology and Physiology del Pilze Puncture and Mycomycetes was written by Anton de Barry. Actually, this book was a German version. That is English translation happens in 1887. So the English name of the book is Morphology and Physiology of Fungi, Mycetiosa and Bacteria, which was written by Anton de Barry. We will know very well about Anton de Barry. He is considered as father of plant pathology, father of experimental plant pathology, father of modern experimental plant pathology. So the next question is gene for gene interaction was studied in the which crop? So gene for gene interaction was studied in the crop linseed rust. So hope we will know that gene for gene interaction was discovered, or gene for gene hypothesis was discovered by H.H. Flor in the year of 1946, not 56, 1946. So it is discovered in the crop linseed. So the next one is Bengal famine occurred in Bengal region during 1942 to 43. That is, Bengal famine is mainly due to, they are asking the causal organism or the pathogen which is responsible for the Bengal famine. That is Bipolaris varizae or Helminthosporium varizae is the fungi which is responsible for Bengal famine. So the next one is the bacterium which produce yellow color pigments or yellow color colonies, that is Xanthomonas, a very important one. The Xanthomonas will produce yellow color pigments or yellow color colonies. That is the identification characteristics of Xanthomonas. You will know that Pseudomonas will produce fluorescent pigments, as like Xanthomonas will produce yellow color colonies or yellow color pigments. So clubroot of cabbage, that is Plasmodioporo brassicae, that enters into the plant cells through. So clubroot of pathogen, that is Plasmodium brassicae pathogen that enters into the plant cells through root hairs. Because if you see the life cycle, so most of the cycle will happens in the root hairs and the cortical cells, except in few stages that happens in the soil water. Later that after when it enters into the root cells and cortical cells, the entire life cycle happens in root cells and cortical cells only, where the hypertrophy and hyperplasia symptoms that we can observe, that is called club root. Root region become clubbing, like bulbing of root region can be seen. That is that tumors or hope you will know that that is due to hypertrophy and hyperplasia. The next one is flag smut of wheat is caused by flag smut of wheat, sorry, Eurocystis triticale, that was introduced into India from which country they're asking. So flag smut of wheat is introduced into India from Australia in the era of 1916. Sometimes they can also ask a causal organism of flag smut of wheat is caused by Eurocystis triticale. And the next one is in pathogen nomenclature, the family ends with suffix. So in nomenclature, the family ends with ACA. The family ends with ACA. So here the next question is the first Indian to collect and identify fungi is K.R. Kritikar. So K.R. Kritikar is an Indian scientist. In 1885. He identified more than 900 types of fungi and he classified it. And he is the first Indian scientist who collected and identified fungi. The next question is first Indian to collect, sorry, the same question that has been asked here, care critical. And the next one is fungicide with both upward and downward translocation in plants. That is versatile aluminum. Most important one is versatile aluminum. And we can also write one more example, metaloxyl. So if both the options has been given in the options, so first of all you can go with versatile aluminum. If versatile aluminum will not be there in the options, then you can go for metaloxyl. So both upward acropetal, downward basipetal. Sometimes they can also ask like this fungicide which having both acropetal and basipetal action or basipetal movement. That is versatile aluminum. I mean metalaxyl also comes under. If the options in options if metalaxyl was the if option versatile was not there, you can go for metaloxyl. The next one is green ear disease survived in the form of oospore. I think we had seen this question already in 2019, I think. And next one is father of modern experimental plant pathology. I told you Anton de Baer is considered as father of plant pathology. Father of experimental plant pathology. Father of modern experimental plant pathology. Everything that is related to Anton de Baer only. So Koch postulates are essential to establish pathogenicity. Koch postulates are otherwise called as pathogenicity rules that we will know very well. So this Koch postulates are used to establish pathogenicity. That's why it's called as pathogenicity rules. Other name of Koch postulates are pathogenicity rules. So the next question is counter strain used in gram staining. Safranin. Safranin is a counter strain which is used in gram staining. The next question is the first edition of Berg's Manual of Determinative Bacteriology, which was published in the year 1923. Here, if it is a determinative bacteriology, Berg's Manual of Determinative Bacteriology, the year is 1923. If it is a Berg's Manual of Systemic Bacteriology, the year is 1984. Determinative bacteriology published in the year 1923, whereas systemic bacteriology published in the year of 1984. Make sure of it. And association between algae and fungi is known as lichens. You know very well about lichens. Lichens are the association between algae and fungi. And the concept of vertical and horizontal resistance was given by Van der Planck. I think in 2019 net question paper itself we had discussed about the concept of vertical resistance, horizontal resistance, monocyclic diseases, polycyclic diseases, AUDPC, vertifolia effect. This all was concepts was demonstrated by Van der Planck only. The next one is common name of Pleurotus florida. That is common name of Pleurotus florida is oyster mushroom. Pleurotus zozarkazu, Pleurotus austriacus. These all are the common name of oyster mushrooms. If you see the name Pleurotus, then you can go with oyster mushroom. The next one is blossom end rot of tomato is caused by calcium deficiency. So blossom end rot of tomato is a abiotic disorder which is caused by calcium deficiency. So the next one is the alternate host for pale millet rust. The alternate host for pale millet rust is brinjal. Brinjal is the alternate host for pale millet rust. The next one is black tip of mango is due to boron deficiency as well as sulfur dioxide toxicity also. If in options boron deficiency will not be there, you can go for sulfur dioxide. If both options will be there, you can go with the boron deficiency, because black tip of mango is mainly due to boron deficiency. The next one is Fusarium wilt. Sugarcane wilt is caused by Fusarium sacchari. So sugarcane wilt pathogen is Fusarium sacchari. Then red onions are resistant to, next question, smudge disease, whereas white onions are susceptible. If you see, red onions are resistant to onion smudge disease, which is caused by Coltrotrichum circinens, whereas white onions are susceptible because presence of phenolic compounds in the red onions, that is catechol and protocatechic acid, which are present in the red onions, whereas these two phenolic compounds are which are absent in the white onions. That's why red onions are resistant to smudge disease. So the next one is Paul Neergard belonging to the field. So we will know that Paul Neergard is considered as father of seed pathology. That's why he belonging to the field seed pathology. So because he is the father of seed pathology. So the sterile hyphae present in the hymenium layer. We will know that the sterile hyphae present in the hymenium layer is paraphysis. So paraphysis is the sterile layer which is present in the hymenium. So the next question is, clamp connection is the characteristic feature of Basidiomycota. We'll know very well about clamp connection as well as dolipore septum. Is the both are the characteristic features of Basidiomycota, whereas crozier formation is the characteristic feature of Ascomycota. Clamp connection and dolipore septum are the characteristic feature of Basidiomycota. Next one is the type of disease which is caused by Tilletia. So the type of disease which is causes the Tilletia is bunt diseases. If you take rust, so many genera which are responsible for the rust. If you take Puccinia, Gymnosporangium, Hemilia, Phragmidium, all the genera which are responsible for the rust. If you take the genera Tilletia, this is only causes bunt diseases in various crops. So the next one is Coliroga disease of areca nut is also known as Mahali disease. Mahali disease of areca nut is otherwise called as Coliroga, or Coliroga is otherwise called as Mahali, which is caused by Phytopthora palmivora. So Coliroga disease of areca nut is caused by Phytopthora palmivora. So the next one is the nature of parasitism of Pythium dibarianum, that is facultative saprophyte. So Pythium dibarianum is a facultative saprophyte. So the next one, the first systemic fungicide which was introduced by, they are asking the scientists who introduced the first systemic fungicide. Van Schemling and Marshall Kulka introduced the first systemic fungicide in the year of 1966. Hope you will know that first systemic fungicide, that is the oxanthin group of fungicide. Oxanthin group of fungicide, that is carboxin, oxycarboxin. These two are oxanthin group of fungicide, and where these two are the first systemic fungicide which was introduced in the year of 1966 by Van Schemling and Marshall Kulka. So the next one is crystalline nature of viruses was demonstrated by W.M. Stanley, because TMB was crystallized by W.M. Stanley in 1935. Hope you will know that which was crystallization was done by Stanley in 1935, where he received Nobel Prize in the year of 1946 for the crystallization of TMB, tobacco mosaic virus. So ELISA was enzyme linked immunosorbent assay, which was developed by Clark and Adams in the year of 1977. So the next one is bendy yellowin mosaic virus is transmitted by the species of whitefly. So whitefly, which act as a vector in transmitting of a bendy yellowin mosaic virus. The species is Bemisia tabasi. So the next one is Indian type culture collection is located at New Delhi. ITCC, Indian type culture collection, which is located at New Delhi. So the next one is the fungi completely used for the genetic studies. So Neurospora is a fungi which is used for the genetic studies, and one other species of fungi is also used in genetic studies, that is Aspergillus. But mainly in fungi, Neurospora is mainly used in genetic studies. If you take bacteria, Agrobacterium is used in genetic studies. If you take virus, tobacco mosaic virus, and cauliflower mosaic virus are the two viruses which are used in genetic studies in particular regarding to viruses. If you take nematode, Chinorhabdis elegans act as a genetic model organism for genetic studies. So they asked here about fungi only. So the model organism for the genetic studies of fungi is Neurospora. If you want the full name, Neurospora crassa, that is a red bread mold. Neurospora crassa, and Aspergillus also act as a, used as a fungi for the genetic studies. So the next one is the formation of naked asci or naked asci is the characteristic feature of hemiascomycetes. So hemiascomycetes characteristic feature is formation of naked asci is the characteristic feature. We can see the naked asci in Taphrin. I hope you will know that. And who reported the transmission of grape fan leaf virus is transmitted by a nematode, that is Ziphinema. Full name is Ziphinema index. So the scientist who reported that the grape fan leaf virus is transmitted by a nematode, that is Ziphinema, that is Hewitt et al. Hewitt is a scientist. He reported that grape fan leaf virus which is transmitted by a nematode, and the nematode name is Ziphinema index. Sometimes they will ask like this: grape fan leaf virus is transmitted by Ziphinema index. Now they had asked like, who reported the grape fan leaf virus that is transmitted by Ziphinema, that is Hewitt et al. So the temperature required for Phytoplasma germination and penetration through zoospore. So for Phytoplasma, 12 degrees centigrade can be required for the penetration of zoospore. For the sporangium production, it will the pathogen required in between 18 to 20 degrees centigrade. Sometimes it will require 20 to 24 degrees centigrade. But in the sense of germination and penetration of zoospores exactly require 12 degrees centigrade, whereas sporangia requires 18 to 24. It may be various, depends on the condition. So the next one is potato scab can be controlled at the pH of 5.2. We'll know very well. Club root of cabbage can be severe at the rate of 5.2 pH, and it can be controlled at the rate of 7.8 pH. How we can raise the soil pH? By the application of lime. If you apply lime, soil pH can be raised. That's why application of lime can be recommended for the control of club root of cabbage. I'm telling, club root of cabbage can be severe at a 5.2 and controlled at 7.8. Whereas in the case of potato scab can be severe at 8 pH, and it can be controlled if the soil pH can be reduced to 5.2. How we can reduce the soil pH? If we apply gypsum, that can reduce the soil pH. So for club root of cabbage, 2.5 tons of application of lime can be required per hectare, whereas in the sense of potato scab, gypsum can be applied at the rate of 900 kilograms per hectare can be applied to reduce the soil pH. To reduce the soil pH, so that the potato scab can be controlled at the rate of 5.2. So the next one is potato scab. I'm the same question again. The potato scab can be controlled at the pH of 5.2. So the next one is genome of mycoplasma. Genome of mycoplasma in the sense phytoplasma. There is nothing to worry. So the phytoplasma contains both the DNA and RNA as a genetic material. Not only phytoplasma but also spiroplasma, that contains both the DNA and RNA as a genetic material. Phytoplasma and spiroplasma both contains DNA and RNA as a genetic material. The next one is expand QTL. QTL in the sense quantitative triad loci. Quantitative triad loci, which was discovered by Gelderman. Actually, this procedure, initially it was set up by Sachs in 1923. I think the procedure was given by Gelderman in 1975, I think so. But the expansion of QTL is quantitative triad loci. So the next one is rice leaf smut is caused by Entelloma varizae. So the next one is Panama wilt of banana is caused by Fusarium. Panama wilt otherwise called as Fusarium wilt. That's why it's called as Panama wilt. So the full name is Fusarium oxysporum pharma species cubensis. Or cubens is the causal organism of Panama Fusarium wilt or Panama wilt. Sometimes it's called as Fusarium wilt. Then test used for the detection of downy mildew of sorghum. Among the given options, sodium hydroxide or soaking test, any wise soaking test can be recommended for the identification and detection of downy mildew of sorghum. So the next one is test used for the detection of downy mildew of sorghum. Sorry, sodium and hydroxide, same question has been given here also. So the next one is the book Biological Control or Biocontrol of Plant Pathogens, which was written by So the book was written by Baker and Cook in the year of 1983, which was a famous book, that the first complete book that is completely devoted to biological control. So before that, there is no complete book that is related to biological control. This is the first book that is completely related to biological control. The book name is Biocontrol in Plant Pathogens. That was written by Baker and Cook. So stem pitting is the characteristic feature that is produced by citrus tristiza virus. So stem pitting is the very common and characteristic that is produced by citrus tristiza virus. The next one is the downward bending of leaves is known as epinasty. Downward bending of leaves is known as epinasty. So the next one, compound which prevents the growth and multiplication of fungi, but it doesn't kill fungi, that is fungistat. Fungicide which kills the fungi, whereas fungistat that doesn't kill the fungi, but it prevents the growth and multiplication of fungi, that is fungistat. The next one is citrus greening is transmitted by Dryophorina citri. I think it is a psyllid. Citrus greening is transmitted by psyllids. That is Dryophorina citri. So the next one is lack or absence of iridospore in wheat stem rust is known as. They are simply asking lack of iridial stage is known as demicyclic. In demicyclic stage of wheat stem rust, there is no iridial stage is present. So if there is no iridial stage, it is called as the demicyclic rust. Lack of iridial stage is known as demicyclic rust. So the next one is name the term used when the disease is prevalent throughout the country, continent or world. Pandemic. So if the disease is which is present throughout the country, throughout the continent, throughout the world, everywhere the disease will be there, it is a pandemic disease. So best example of pandemic disease, late blight of potato and wheat stem rust. These two can be we can call it as a pandemic disease, because this disease will be there everywhere. That's why it's called as ubiquitous, in the sense found everywhere in the world. Wherever the corner you go on the planet, you can find this disease. That's why it's called as pandemic. And the next one is crescent phase can be observed. Hope we will know very well about, from UG itself we are studying crescent phase can be clearly observed in bacterial blight of rice, most destructive phase of bacterial leaf blight disease. So crescent phase can be observed in bacterial blight of rice, which is caused by Xanthomonas vorizae, pathover vorizae. The selective medium for the isolation of Pseudomonas. So Kingsby medium is a selective medium for the isolation of Pseudomonas. The next one is autoclave sterilization by heating at the rate of 15 psi for 15 minutes. At what temperature they're asking? So minimum, hope you will know that basic temperature for autoclave is 121.6 degrees centigrade for 15 minutes for 15 psi pressure. So the next one is, in which disease of sorghum a long columella is present? That is in which disease of sorghum a long columella is present at the center of each zone, that is a loose smut. In a loose smut, long columella is present. Remaining grain smut, head smut, and long smut, we can only observe the small columella. So we can hope you will know that there are four sorghum diseases, the main important disease, that is sorghum rust diseases. That is grain smut, sorghum grain smut, head smut, loose smut, and wheat smut. Among these four smuts, loose smut will produce a long columella. In nomenclature of fungi, order ends with, in nomenclature of fungi, order always ends with else. And if you, next question is black rough encrustations on the surface is the characteristic feature of black heart of potato. If you take the black heart of potato, a rough corky outgrowths or rough corky surface can be clearly observed. If we touch, we can clearly sense it. That is due to black heart of potato. Next one is sporangiospores are dichotomously branched. Don't think that if you found this dichotomously branched, it's immediately grow for pernospora or something else. It's not true. So we can study the question clearly till end, then only you can understand the question completely, then you can go for answer. Don't go it immediately, because I had done the same mistake for many times. So the sporangiospore is dichotomously branched at acute angle and tips are branched or expanded with cup shaped branches and sterigma present. Note this point, with cup shaped branches and sterigma present. The cup shaped branches we can only see in Bremia only. So mostly Pernospora and Bremia almost characters are resembles, but the main differentiation in Bremia is cup shaped sterigma can be observed. Cup shaped branches with sterigma can be observed in Bremia, whereas it absent in Pernospora. The next one is dispersal of plant pathogens through animals is known as zoochory. So the dispersal of plant pathogen through animals is known as zoochory. If it transmitted by air, it is called anemochory, and any birds ornithochory, and some other will be there. If it is transmitted by humans, arthropochory. So hope you will know that. So the next one is the secondary spread of Phomopsis blight of brinjal can takes place through conidia. So secondary spread of Phomopsis blight of brinjal can takes place through conidia. Hope you know about the causal organism of Phomopsis blight, which is caused by Phomopsis vexens. So which nematode has a significant domestic quarantine in India? That is a golden cyst nematode, and one more disease is a fungal disease, that is potato wart, which is caused by Synchytrium endobioticum. So the nematode they only asked the nematode which has a significant domestic role in quarantine in India, that is golden cyst nematode. So decanary system of nomenclature of wheat stem rust was given by, so decanary system we can see that was given by Nagarajan for wheat stem rust. So the book Disease Resistance in Plants was written by Vander Planck, and he written 5 books: Plant Disease Epidemics and Control, Genetics of Plant Disease Resistance, and Disease Resistant Plants, and another 2 books also there. But the famous book is Plant Disease Epidemics and Control, which was released in the year 1963. So the next one is the National Research Center or Integrated Pest Management (NCIPM) is located at New Delhi. So National Research Center for Integrated Pest Management is located at New Delhi. So the next question is antifungal antibiotic was developed by Tirumulachur. What is that antifungal antibiotic name they are asking? As I told you earlier, the antifungal antibiotic is Ariofungin, Dermastatin, Hyamycin, MYC4. These all are the antifungal antibiotics which were developed by Tirumulachur. He is considered as father of Indian antibiotics. I am telling again, Ariofungin, Dermastatin, MYC4, and one more will be the actually forgot Ariofungin, Dermastatin, MSC4, and other other antifungal antibiotics also there. These are the important one that was developed by Tirumulachar. And the last one is father of soil solarization. Jacob Cotton is considered as father of soil solarization. He is a Israel scientist. He's the only father that is still alive in Israel. So regarding other fields, all the fathers are died so far. So the only father still alive is Jacob Cotton. He is considered as father of soil solarization. So these all answers are exactly based on the options given in the exam, and the above questions are purely based on the memory. So this is ASRB NET 2006 question paper regarding plant pathology. This is a collective work from Team Geeky Researchers.